So what I'll do is talk to you about the genetic analysis, I guess, platform that we're developing for looking for resistance factors to rose-rosette disease, black spot, of course, and any other diseases that we work with, and cercospora leaf spot being the other one. We're also doing a variety trials in Texas where we're looking at black spot, cercospora, particularly with respect to diseases, and of course in Tennessee and Oklahoma they'll do rose-rosette disease. But this is again complementary to that work as well. We do have a variety trial here. We have a variety trial in Tennessee, and we have another one in Oklahoma. The genotypes are the same. Slight differences in makeup depending on what survived where, but we treat it as being the same panel. This is a graph that I normally show to show you what I mean by the platform we built to do genetic analysis in tetraploid roses. And again, this is something that you could have not thought about doing 10 years ago because a lot of the technology and computational tools were not available to do this so readily now. And again, we do quite a bit of mapping, and we use MapPoly and PolyMapR. We've developed a tool called Qploidy, so we can actually run genetic data from any rows, and we can tell you whether it's a diploid, triploid, tetraploid, pentaploid, even if it's a hexaploid. And we've identified genotypes that have ploidy levels higher than 5 in some of the samples we looked at. So the reason we do that is if we know the ploidy, then we know how to handle that genotype for genetic analysis or any downstream analysis thereafter. We take genotypic data that we generate mostly from the Axiom array. We've done some rat sequencing not very successfully, but we use the Axiom array, process that data, and then we try to combine it with phenotypic data from field trials, and then combine those to do QTL analysis, looking for factors that govern a particular trait, for example. We're starting to move into genomic prediction, which is another field where you use genotypic and phenotypic data to develop models that can predict a phenotype based on a genotype. That's why it's called genomic prediction. And we're exploring how to apply it to tetraploid rose breeding as well. And again, you've seen that a lot of our focus has been describing some of the QTL mapping that we've been doing. We've started with mapping populations, and we are little by little moving towards germplasm collections of varieties, breeding lines, species, and what have you. So mapping populations are the product of crossing two varieties or two accessions. So the genetic diversity segregating in that population traces back to two parents. But if we look at germplasm collections, we're looking at more broad array of diversity. So hopefully we'll find more things than we can find in mapping populations. Therefore, we're moving from mapping populations to germplasm collections little by little, as we learn how to do that better based on the tools that are available. As I said before, we're part of the variety trials team. And here we've got a couple of pictures of us visiting the folks in Oklahoma and Tennessee. And again, the idea is to look at about 200 plus accessions that were replicated at least 5 times. Less in some areas, but we try to go for 5. And again, they're being grown in Somerville, Texas, which is Hortrek. We will take you there later. They're in Crosville, Tennessee, and also in South Tulsa or North Bixby, and I just call it Bixby. In any case, again in Texas, we place mostly black spot and cercospora, although we phenotype for a number of other traits as well. Some are ornamental in value. Tennessee, mostly rose-rosette disease, although we've gone there to actually do black spot and cercospora, and that's why we visited the teams in Oklahoma and Tennessee this summer. We picked up, we scored for black spot, cercospora, defoliation, and flower intensity actually while we were visiting them. And again, it's great to see the material in other locations besides ours as well. This is just a quick summary of what the data for those traits is, and I have black spot on top, then cercospora leaf spot, and then defoliation, This would be the 2 years of data that we have for Texas, August in Tennessee, and September in Oklahoma. And you can see there that you can clearly see there's G by E. The collection of roses react differently to different environments with respect to different traits. But another thing that you'll see there is there's a good range of resistant to susceptible materials for every given trait in the case of diseases. And of course, there's a range also with respect to how much defoliation those genotypes endure or are subjected to. Okay. We're in the process of cleaning up the data before we do a more formal analysis, but I've looked at some of the heritabilities or repeatabilities, and they look pretty good. So this again will be something that is feeding the GWAS work that Reswan is doing, is as we get more data from more traits, process this with, and he'll run it through his pipeline to do QTL detection work. Similarly, I want to take the phenotypic data and the genomic data that we already have and develop genomic prediction models for each of those traits as well. And I'll explain to you more or less how we utilize that later. So we've got mapping populations that Navdeep described to you. And again, the goal there is to identify factors for resistance to rosette disease and black spot particularly, although we do get into other traits depending on whether or not we have the data. You can think about it as a population of about 820 individuals that is interconnected by specific process. So we use that for looking for QTL, but again, we want to then utilize that for genomic prediction modeling down the line as well. And we have here our handsome collaborators at the bottom, Nar, Francisco, and Nectar. And we value very much their, I guess, collaboration to get us access to other environments with mapping populations that are planted, again, in Texas, Tennessee, and Oklahoma as well. All 3 locations have those populations. Similarly, Reswan talked to you about the GWAS panel. I know the numbers of what he showed and what I'm showing are slightly different depending on whether we want to count this or that genotype in any given case, but between 2024 and 2025, we've put together a collection of about 487 individuals, about 200 or so from the variety trial, and then we put an extended set of varieties and breeding lines that we call the GWAS. So what we were trying to do is to, instead of just working with 200 individuals throughout this project, we would work with something close to about 450, we're close to about 487 individuals. And we wanted to do this to increase the power of QTL detection of the panel. The more individuals you have, the better off you are to do it. That means that then we've got about 480 to 500 individuals that we could also use for genomic prediction model development. We also had variety trials that were grown previously. There were about 345 or so. And you can see those guys in this other circle here versus that one. And then, of course, this is the area of overlap between them. It's because there are these overlapping genotypes between these 2 experiments that was separated by time that we can actually interconnect them computationally so that our population size is a lot larger than individually, if you think about it that way. Again, these materials are planted in multiple locations, Texas, Tennessee, and some of them in Oklahoma. Okay, and then data from those locations then can be applied to this type of an analysis. Again, this can be used for genomic prediction down the line as well. Now I'm going to talk about QTL to make a point. So we've got mapping populations that Natif talked about. We've got the, I guess, GWAS plus variety trial material that Reswan and Zina have been working on. And this is a kind of a summary of the QTL they've all found relatively recently. And, you know, you can assume maybe this QTL is the same, maybe they're all different. But if you count them as being different, then, well, we picked up something close to 24 QTL for black spot and circus put together, right? So anytime we look for QTL, we find them. Sometimes we find things that we think we found before, but we're also finding things that we never found before. If you remember the meta QTL analysis work that Tessa presented yesterday, her research suggests that we may have up to 46 QTL if you look at the 7 from rososette disease, 14 from black spot, and another 25 from cercospora. Again, now we're up to 46 total QTL. So maybe these 46 QTL are not all that different, or they may be different from the ones that we have recently found. So we could say maybe we're up to 60-something QTL, if that's the way you want to look at it. Maybe some are the same, maybe they're not. In any case, the picture here is that the resistance to these diseases is complex. Some are more complex than others, but they are complex, meaning that they're controlled by multiple factors working together rather than, major genes at this point. There may be some genes for immunity to rososette disease, but I don't think we have found them. There may be genes that are supposed to provide strong resistance to black spot, but I don't think we found them. But what we're finding is that we've got multiple factors that together to provide quite good protective, power against some of these diseases. And these are quantitative factors, not qualitative ones. And that is the direction we want to go for this specific reason. So in the past, we thought that traits were controlled by many genes of small effects. Then we started doing QTL mapping, and then we identified factors that actually could explain quite a bit of the variability. You would run a QTL analysis, and then you would find 3 factors, each of those explaining between 10 to 40% of the phenotypic variance. And you say, this is interesting. If I can explain that much of the variability, I might have major genes that actually explain a quantitative trait. So we started thinking that a lot of the traits were controlled by oligogenic systems, meaning that few genes of large effects were behind quantitative variation. And the problem is that the way QTL mapping works, it is an experimental setup that creates the conditions so you can pick up factors that may not have a strong effect, but they look like it in that mapping population. They look like big guys, but when you move them out of the environment they're in, they're not as big as they seem, meaning that they're really small effect loci compared to the rest of the genome. Then we decided to thinking, I think we're going back to the polygenic model where quantitative traits are controlled by many factors of small effects, not a few factors of large effects. And that's where we are now. But for most traits, we think quantitative, variation is controlled by the action of many genes of small effects. So that means that if we want to work with black spot, we want to work with cercospora, and we want to work with rososette disease, we need to be able to handle many factors of small effects in a breeding context, if that's the purpose. And we cannot attempt to do what's called marker-assisted selection, where if we have specific factors that are tagged by a particular marker, and we want to move them from a donor to a receptor, and hope that by growing a large population, identify an individual that has inherited that QTL, you run into a numbers problem. And this is what this busy slide shows. This is the equation to calculate what is the minimum population size to obtain a segregant of a particular genetic architecture. So if you want a pyramid or put together 5 genes, you could do that in 145 individuals, meaning that if you grew at least 200, at least 1 or 2 would have those 5 genes transferred from one donor to a receptor. If you had 10, you need 4,000 individuals. If you have 15, you need 150,000 individuals. So you can see how many QTL do we have so far? 46, so it's going to be huge. This is a logarithmic scale. So this is only 15 genes that we want to try to pyramid together. So trying to put things together that way is going to be virtually impossible. We just cannot handle population size that large. Now, the theory now is that you can have 2 genotypes of very comparable levels of resistance if they contain the same sets of favorable alleles, or maybe different sets that are complementary. You could have one that is resistance for a number of genes, another one resistance for another set. If they're complementary and you cross them, you can get progenies that are going to be better or worse. If they are the same, you're going to make a cross, and the progeny are going to be just as the same as parents were. All right, so you can work with the same allele or different alleles as long as they're favorable. And that's what the genomic prediction approach allows you to do. One concept that I like to drill down is the concept of breeding values. This is a concept that was born in the cattle, I guess, arena, where there's a really great example, is that the value of the semen of a bull for milk production is based on how well the descendants or the relatives of that bull perform with respect to milk production. We can obviously cannot harvest milk from a bull. You can try, but I'm sure you're not going to be successful. So how is it that you know how good is that bull to producing progeny that produces good, milking cows? But again, you gauge the value of that bull by how good his sisters yielded milk, the mother of that bull yielded milk, and of course, daughters of that bull yielded milk. And if you understand the relationship between them, you can assign a breeding value to that bull. We can do the same thing in roses if we know the relationship of one rose to another. We can gauge the value of a rose by its relationship to other roses and how good those other roses were or are. And that's how breeding values are applied in cattle breeding. And now that is a concept that is definitely using in large-scale breeding programs in the US for row crops particularly. And that is something that we were trying to do. So we want to use breeding values to assess whether a genotype is good or bad for a particular trait. I'm going to show you equations. Don't have to worry about them. But if you understand them, then great. So the breeding value traditionally was done through pedigree, understanding the pedigree of lines. Now, if we have perfect pedigrees, then we can do the same thing with roses. But if we don't, we can use genetic relationships based on markers to be as a proxy for that pedigree understanding. But in the end, there's a couple of really smart people in a paper published in 2001, where they showed through mathematics that a breeding value, if you talk about markers in a genome, is really the sum of all the allelic effects that you can attribute to every marker. So you have thousands of loci or markers in the genome, and you can tell me what is the effect that each of those has on the phenotype, be it positive, negative, or neutral. If you sum them, then you get a breeding value. Okay, and that's what this formula shows, the summation of all of the marker alleles and their U, which is their effect. So if we know the effect of every allele and how it affects any given trait, and we know that throughout the genome, then we can calculate a breeding value for any plant as long as we have the genotype for that plant. And if the breeding value is high, it's great. It has high genetic merit. The breeding value is low, it has low genetic merit. We don't want that individual. Okay? And again, this is how genomic prediction then works. You've got a genotype, you have a model with effects, then you can predict that phenotype. And then you can make selections based on genetic data if that's what you want to do. All right? So then the good thing about this is that we already know that the traits that we work with have complex inheritance. It's going to require multiple factors, not just one or two. Then this is a method that basically scans the entire genome and tracks the entire genome to give you those breeding values that you want. So you can work with complexity in a very, I guess, relatively well-managed way. This is work by Jikan Lau, that was mentioned earlier, was a former postdoc in the program. He works at USDARS, and he was the first one to do genomic prediction in roses, as far as I know. And he took data from the populations that we had at that time. They were biparentals. he had genotypic data, phenotypic data. So he calculated breeding values and then we used a weighted selection index to identify the best performing lines based on a combination of traits. These are the 4 populations that we went back to. You've probably seen bright eyes by my girl, stormy weather by bright eyes, probably brought up earlier. Then the WB and WC different populations, and we've identified the top 100 or 50 lines from each of those populations that, in our view, contain the highest frequency of favorable alleles for, again, in that case was, sorry about that. So we were looking at rose-dusty disease, black spot, cercospora, defoliation, and flowering intensity. So for each of those traits, we calculated a breeding value for every plant. Then we normalized those and then added weights based on relative importance. Then we sum across that collection of breeding values, if we will, to get an index number, that is this I in the formula. And the individual that has the highest I guess, is the best one, has the best balance of phenotypes, he's got the best balance of genetics for all of those traits. If the I is low, then it's got the worst combination of alleles for each of those traits. So that's what we have. So one of the outputs from this, if you think about it, it's not a prediction, I guess, formula or model, but you can actually look into individuals for which you have phenotypes and genotypes and pick up the ones that are the best for that particular trait. Then this is what we call parental selection, if we want to use them in a breeding program down the line. Or you can decide to go ahead and promote those as varieties for release subsequently before testing. Okay, so we've got some of those materials already selected, and you may or may not see them in the field today. So now that we have this project, the idea is to take 820 individuals from these 6 biparentals that we have, the 743, I guess, accumulated, curated over time, and then really put them together to produce a panel of about, I don't know, close to 1,600 individuals or more to try to develop a more robust, I guess, genomic prediction model to, again, select lines through an index that are the best out there, or to develop a prediction model to be able to do sibling selections down the line, or for other applications. So that is the genomic prediction, I guess, platform that we're now building. So we've got now the ability to genotype, phenotype, do a lot of genetic analysis through a number of software. Now we want to build a genomic prediction layer to that analytical pipeline that we have so far built. So the top part is how you develop a prediction model, basically a germplasm that we call a training population. We genotype them, we collect data, and then we develop a model, and then that model is deployed where you have now breeding populations, go through the model, estimate genomic estimated breeding values, then use those to select a subset that go for testing and release, or go back to the training population, or go back to the breeding population. So that's basically how we want to use this sort of platform. And again, should allow us to manage quantitative traits better than just marker-assisted selection. Okay, so this is the stuff that we have, and some of the applications of that platform that we're trying to build, or we're building little by little right now. And again, the RES1 and Xena will be part of that endeavor. This is my summary slide. So we are identifying a number of genes and QTL are behind a number of traits. So disease resistance and sometimes ornamental. We've noted that the inheritance of those are more and more complex as the more we look at them, and we're moving to find genomic prediction approaches to manage that complexity a little bit more effectively. We want to identify germplasm that it has good genetic merit for breeding. We could also use the same approach to promote material for testing down the line. All right, I'd like to thank a lot of folks from from the team. And I'm showing you photos of Jennifer, who you now know, is taking a new role as project coordinator. Pam and Stacy have been out there helping Jennifer take care of things, but the three are the ones that really keep our roses well kept and managed at Hortic that you'll see today. And there's a number of other researchers and graduate students and former graduate students, postdocs and former postdocs that are the ones that actually get to work done. Thank you to our collaborators, all of you in the project, and of course the industry that supports the project as well. All right, I'll stop there. Thank you. And I think we have some time for questions. Yes, go ahead. We have a few minutes. Go ahead.
So under this scenario of writing release, would that be under the office of the public domain or royalties? How would that?
So yeah, that's a good question, right? I guess the thing that we would like to do is be able to have some sort of a cash flow so that we can maintain a breeding program. I mean, it's expensive to have people phenotyping, it's expensive to do a number of things. Just growing roses itself is extremely costly for us. So we definitely need to have a mechanism to feed money back into the program. The way they do it traditionally in plant breeding programs in the public sector is they do releases that are subject to royalty revenue. And part of the royalties, of course, is going to stay up there somewhere, and hopefully some will trickle down to us as well. So that will be the scenario, the formula that we would like to pursue.
Question. Dr. Oscar, you mentioned that Dr. Liu did the genomic prediction for roses first time.
Dr. Lau.
Dr. Lau. Can we know what the prediction accuracy was?
Well, again, you are mapping populations. So the accuracy means nothing because the equation only applies to that mapping population. But since you're using performance per se and the genetic relationship of the genotypes to each other, you can calculate what's called a GBLUP, which is again a genomic prediction output that would allow you to differentiate the individuals that are not only good phenotypically, but also because they have the largest amount of favorable alleles. Then you say these are the best that I have from that population. So it's a parent selection application of genomic prediction theory.
Does it matter if I genotype my parental population or my biparental population for my prediction?
So again, for the prediction, what we're trying to do is build this, right? In order to have a robust prediction model that is more generally applied, you need to look at a much broader array of materials that could also include the mapping populations. So we need to build what's called a training population that I show in the next example here. So we need to build this guy here, which will be composed of specific varieties, not all of them. We may or may not have rose species in there. We may or may not have mapping population parents or members, but it'll be the optimal population that generates a prediction model that is robust to predict down the line that would have high accuracy for the traits that you're trying to predict.
Last question. Has genomic selection done in roses? Yeah.
Well, we have done it, but the way I described it, by selecting individuals that have the highest frequency of favorable alleles for multiple traits, yes.
You're moving to the next stage. Like you've evaluated it in the field later.
We have segregated the material and we're going to continue testing them. Yes. So we've applied this, right? But you can't say that we've actually done something that has been commercially successful because of that activity. That has not happened yet. Any more questions? If not, I guess we could.
Yes, I have a question.
Please go ahead.
Hi, I'm Bruce Monroe. I'm with the American Rose Society Research Trust. We're sort of a mini micro funding agency. We funded Dr. Wyndham's original work when nobody else was interested in rose rosette disease. But I've listened to all this excellent research. It's involved a lot of hard work and a lot of both field work and work in the laboratory. But as a funding agency, even though I'm a minor funding agency, and I also grow roses and work with the public garden as a potential customer, let me ask this general question to the group. How long before I can go to the local garden center and buy a rose that is resistant to rose rosette disease?
Come on, guys, provide some answers.
Funding agencies will
That's
ask
a
that
very
question.
fundamental question, but I think the issue that we're seeing so far is that we haven't identified very good sources of resistance to rose rosette that you could say have large effects that could be quickly, I guess, deployed using a market-assisted selection type approach. You would have that very quickly. Now, if all of the resistance that we find or that we are able to utilize happens to be those that are controlled by many factors of small effects, that's going to take a process that can actually accumulate them fast enough so that you can deploy genotypes that have fairly good levels of resistance equivalent to major gene resistance. And there's a lot of work in wheat, I guess, rusts, where they are able to deploy quantitative resistance that has been built in such a way that it resembles major gene resistance that has been normally utilized, but major gene resistance is known to be not long-lived in that it breaks down within 3 to 5 years. So if we take major gene resistant material out in roses that has resistance to rose rosette disease, that major gene resistance is going to last, I don't know, maybe 5 years. And then after that's going to break down and we're going to go back to square one to build it again. I think we need to take quantitative resistance and push it forward with this genomic prediction technology. So we build durable resistances to all these diseases rather than major gene resistances, which are the easy ones to do, but the ones that break down fastest as well.
I'll make a comment on that too. To answer your question, you could probably go out there and find a resistant plant. The problem is we cannot predict with good accuracy which one and what sort of virus that you might have in your region and what it would interact with. So to say a different question is how long before we learn some of those things that is deployable. And I think that's what Oscar answered. It's it's trying to find those things, and then what's the mechanism to input, and how long are you willing to?
Sustain the effort.
Yes.
Yeah.
Go ahead.
Go ahead.
At this point, what is the dream method that you need to penalize?
I would need a genotyping platform that I can do $5 per sample, not 90. That's my problem right now. It's very expensive to genotype. Even though they tell you with sequencing prices going to the floor and this and that, your price per data point is so cents of the dollar. Yes, but the price of processing a sample is not going down, it's going up. I get more information per sample, yes, but each sample costs me, used to cost 50, then 60, then 70, then 80, now 90%. So that's what I need is a cheaper, high throughput genotyping platform for roses. And again, we are among the few labs that work on roses in the public sector who are the folks that should be developing those resources to be able to do the things that we want to do. But we are a small community and we don't have the economic attraction that larger communities have to companies that develop these technologies. And I've been working with Thermo Fisher Scientific. We were developing a sequencing-based platform for roses. And then one day they call me and they say, we decided to scrap that technology. We're not going to do that anymore because we can't make money. So we stopped it. So we're back to just a race. That's the only platform we have so far.
Do you have to create array each time, or one-time array? Can you use it?
Arrays have to be synthesized. So the arrays are being designed, but they have to be synthesized for application and use. It's like a flow 7 sequence. You got to make a new one to do a sequence.
Bruce, you still have your hands raised. Is there any other comments you would like to make?
Why do we know that the virus is specific to roses? What is it in the closely related genera that cause them to be resistant? It apparently doesn't affect apples. Now, is it just that the mite doesn't affect apples, or there's no vector to spread it, or is our apples really resistant? Maybe we should be looking and see why other closely related genera are resistant and see what kind of resistance they have.
So non-host resistance has been another strategy that has been explored, but the problem is you want a rose, you don't want a plume. You want a rose, you don't want a peach. You want a rose, you don't want a blackberry. So to turn rose into a non-host, you have to change the rose to something else, which is, I guess, kind of it just kind of, to me, it doesn't make sense. Pathogens do jump from one species to another. That's not unheard of, but using non-host resistance to engineer resistance on a species, I don't think has been successful yet.
We certainly don't want to change the virus so it attacks apples. That would be a disaster.
That can happen faster than the other thing around. Yeah. All that has to happen is that Philocopters decide to like peaches, I guess. Something like that. Yeah. So I don't know if you know the story about Fusarium head blight in the Midwest. Fusarium was never a disease of Fusarium head blight was not a disease of wheat until corn acreages started creeping into wheat acreages in the Midwest and the Great Plains. So the cohabitation or the commingling between corn and wheat made Fusarium jump into wheat, and they became the most important disease of wheat in the country. And there was no resistance in wheat to that new pathogen ever. So the US developed an initiative, a granting program to promote breeding and searching for resistances, etc., etc. 30 years later, we got resistant varieties to Fusarium head blight. That's what it takes to fight some of these problems. Concerted efforts, organization, and support and money to do this right.